Let's look at the role of calcium in muscle contraction. So what this figure is showing is the sarcomere in a more of a relaxed state on this side, and then the sarcomere in more of a contracted state on the right here. So notice the difference. When muscles are in a relaxed state, calcium is stored within the sarcoplasmic reticulum. So here we see all these little small dots represent calcium ions. And if the calcium is in essence sequestered and stored within the sarcoplasmic reticulum, there's no calcium to bind to troponin on the thin myofilament. After stimulation by a neuron, which we'll be discussing later, electrical impulses or action potentials travel along the sarcolemma, which is the plasma membrane of the muscle cell, and travel down deep into the muscle cell using what's called transverse tubules or T-tubules. What this does is it gets the electrical current, the action potentials, closer to the sarcoplasmic reticulum. And within the sarcoplasmic reticulum, there are little channels that are stimulated to open in response to these action potentials. And when those channels open, calcium is then released from the sarcoplasmic reticulum. It will then go and bind to the troponin, which is on the thin filament. So here's the thin filament showing the troponin here in blue, the calcium shown here in purple. When calcium binds to the troponin, that is going to move tropomyosin, which you remember, tropomyosin is the blocker, but it has moved out of the way, and it allows the myosin to bind to the actin. So to best understand the roles of calcium, the sarcoplasmic reticulum, transverse tubules, troponin, and tropomyosin, what you can do is just compare a relaxed state versus a contracted state. The next few slides are all kind of a review. This table discusses the different components of a sarcomere. On the right here you see two sarcomeres held together with a Z-disc. Here they are in a relaxed state. Here they are in a partially contracted state, and then here's when the muscle is maximally contracted. The table just discusses the different components of a sarcomere. The Z-discs, the A bands, which are wherever you have overlapping thick and thin filaments, and it gives more of a darker pattern in the striated muscle. The I band is the lighter region, and it's lighter because there are only thin filaments located at the I bands. So only thin filaments, no thick filaments. So looking at this drawing, here we see the I bands in the relaxed, partially, and then in a contracted state, the I bands pretty much go away. The H zone is found in the center of every A band. So if the A band extends from here to here, and it is the darker band, within that A band, there's often referred to as the H zone. And the H zone also disappears as the muscle contracts, but that H zone is where you only have thick filaments. And then finally, the M line we mentioned previously, that's a structural protein called myomesin. That's found in the central region of the A band, and that again helps stabilize the thick filaments. This is a drawing, a schematic, putting kind of everything together that we've talked about, a lot of different components inside this muscle cell. We can go through each one of them, and you can see again how they are involved. But the outer layer is the sarcolemma, that's the plasma membrane. At some point, this cell is going to be stimulated by a neuron. That's going to cause an electrical current to travel along the sarcolemma, down the transverse tubule, so we can release these calcium ions from the sarcoplasmic reticulum. The calcium ions would then go and diffuse into the myofibril, made up of sarcomeres, attach to the troponin, move the tropomyosin, allow the actin-myosin cross bridges to form. Then a big requirement is ATP. So there's a number of structures in here used to generate large amounts of ATP, like the mitochondrion, the myoglobin, which is going to supply the oxygen for cellular respiration, the glycogen granules or glycogen molecules that will supply the glucose for energy production. And as long as you have enough energy, these sarcomeres will contract. If you've got many sarcomeres found within myofibrils, the myofibrils will contract, and then they have to be tethered to the two ends of the muscle cell using the protein called dystrophin. So dystrophin helps attach these long rods to the ends of the cell, as well as some of the extracellular material. So you can imagine if you have a genetic defect in dystrophin, and we call that muscular dystrophy, these myofibrils aren't tethered very strongly to the sarcolemma of the muscle cell. This is a table summarizing all the proteins we've talked about. I'm not going to read this table to you, but you can go through, and again, you notice that in the world of muscle proteins, you have contractile proteins, myosin and actin, regulatory proteins, tropomyosin and troponin, and then structural proteins that one way or another help arrange the thin and thick myofilaments into sarcomeres, help stabilize the sarcomere, or help attach the myofibrils and reinforce their attachment to the cell membrane. So you may want to go through these and read these descriptions once again in order to keep your skeletal muscle fiber proteins straight. The next video is going to discuss what we call the neuromuscular junction, which is the junction between a neuron that extends from your brain or spinal cord to the muscle cell.